Hi, Assalamualaikum. My name is Nadia Binti Murtadza from Bachelor of Science Biology UiTM Perlis. I would like to present my final year project, The Potential of Zingiber Extract in Antibacterial and Antioxidant Activities. Zingiber is one of the Zingibraceae species, the largest families of plant kingdom. The local name is known as lempoyang or bitter ginger. Various ginger of the Zingibraceae family is widely used in medicines in Southeast Asia because of their antibacterial and antioxidant properties. This study will focus on the extract of rhizomes to analyze the antibacterial activity by using methanol solution against a diverse range of organisms comprising gram-positive B. lichen and gram-negative E. coli bacteria. The purpose of this study also to detect antioxidant activities by using DPPH radical scavenging assay. Why Zingiber extract was chosen in my research? Among all the natural goods, Zingiber is still undervalued. Ginger is widely used as a common ingredient in cooking. Instead, this lempoyang species does not contribute to the food industry because of the taste itself. The rhizomes should be used lightly and according to personal taste, as the bitter flavor can become disagreeable if taken in large quantities. Furthermore, the plant can be found naturally scattered in damp and shady areas such as near rivers, waterfalls, and other water sources. The importance of Zingiber from a commercial perspective, it is a medicinal plant with great potential for cultivation that require low cost. Next, Zingiber plant has been ranked 14th under 18 identified herb species proposed to be commercialized by Malaysian government due to its high value market. Furthermore, this species is easy to purchase nowadays from websites or e-commerce platforms such as Shopee. By so many values exist in Zingiber community need to be conveyed to start cultivating this plant to ensure the survival of this plant and protect the species from extinct. The objectives of the research are firstly to extract the rhizomes of Zingiber by methanol extraction. Secondly, to evaluate antibacterial activities of Zingiber extract against pathogenic gram-positive B. lichen and gram-negative E. coli using this diffusion method. And thirdly, to analyze antioxidant activities of Zingiber extract using DPPH radical scavenging assay. So, what is antibacterial agent? It is a substance that can kill microorganisms. The antibacterial activity of extracts from the rhizome of Zingiber is related to the previously mentioned bioactive compounds, which can attack cell wall and cell membrane, affecting permeability and interfering in cellular functions such as electron transportation, nutrients, synthesis of proteins and nucleic acids, and also enzymatic activity. Thus, such bioactive compounds may have several invasive targets that may lead to the inhibition of microbial growth. Next, what is antioxidant? Antioxidants is a compound that inhibits oxidation, a chemical reaction that can produce free radicals and chain reactions that may damage the cell of organisms. As example, in our body, in human body, are exposed to various toxic chemicals such as form of cosmetics, food, lifestyle, or harmful radiation. This will release free radicals that is highly unstable and harmful for the body. In order to stop the free radical process, some compound have to donate an electron for these free radicals so that it becomes stable. These compounds are called as antioxidant. In this diffusion method, firstly, sterile 6 mm diameter filter paper discs were impregnated with 10 microliter dilution of different concentration of rhizome extract, that is 10%, 30%, and 50%. Ampicillin act as positive control and 5% of DMSO act as negative control. For the antioxidant activity, ascorbic acid and methanol were mixed to compare the antioxidant activity. In this study, ascorbic acid acted as standard. Absorbance readings were taken in triplicate after 30 minutes using spectrophotometer at 517 nanometer wavelength. The table shows the results of the maceration method. There were 1000 grams of fresh rhizomes of the plant. After drying process, mass of the dried rhizomes is 112.22 grams. The differences of mass between fresh samples and dried samples are due to loss of water during drying process. In the research paper, methanol was used as a solvent as methanol is the most commonly used extraction solvent due to its high polarity, which could produce high extraction yields. Next, for the results of antibacterial activity, the observation of inhibition zone is to study how well the rhizome's root extract resists bacteria. Ampicillin susceptibility disc, which is one of the antibiotics, was used as a positive control, while 5% of dimethyl sulfide DMSO was used as negative control. 5% of DMSO was used because it enables an easy solubilization of the extracted compounds. In the results of antibacterial activity, the observation of inhibition zone is to study how well the rhizome's root extract resists bacteria. Ampicillin susceptibility disc, which is one of the antibiotics, was used as a positive control, while 5% of dimethyl DMSO was used as negative control. 5% of DMSO was used because it enables an easy solubilization of the extracted compounds. From the results, we can conclude that B. lichen are susceptible compared to E. coli. Susceptible means the rhizome extract are capable to kill the bacteria, while in E. coli bacteria are resistant. This is because gram-negative have a lipopolysaccharide membrane. The graph shows the results of antibacterial activity between plant extract and ascorbic acid. The antioxidant activity is expressed by IC50 value, which is defined as the effective concentration of substrate that causes 50% loss of the DPPH activity. Based on the study, the IC50 value for zingiber-zurumbat rhizome extract is less compared to the value of standard ascorbic acid attained in the experiment. However, the plant still has shown good antioxidant activity. As a conclusion, based on the results from the study that have been conducted, zingiber-zurumbat was successfully extracted by methanol solvent. Next, the antibacterial activity of the methanol extract demonstrates the highest inhibitory zone occurs on B. lichen. And lastly, antioxidant activities of zingiber-zurumbat extract using DPPH radical scavenging assay has been analyzed. For recommendation, further studies might be done to analyze more about the biological activities for zingiber-zurumbat because the plant is underrated and do not get much attention as other unique plants. Besides, the next studies should be more focused on each part of plants. It might give more details and data for every part, which could have greater results for biological activities. Regarding extraction, reflux extraction is the most employed technique for preparative separation. Thank you.